Students, we will start with the new chapter, Principles and Processes of Biotechnology. So first we will understand what is biotechnology. Biotechnology is the branch of science which deals with the use of advanced tools and techniques in biological forms to produce useful products and processes to human beings. It means if in any way it will be beneficial to human being or any animal or plant or our environment. And we used to employ several advanced tools and techniques, and we utilize the life forms to manipulate the things and to get several useful products. So first we will see what are the principles of biotechnology. So there are two main techniques, or we can say principles of biotechnology. First is genetic engineering and second is bioprocess engineering. So first we will see what is the meaning of genetic engineering. When we used to employ several techniques to manipulate the genetic material, either it could be DNA or RNA, and to introduce the manipulated DNA into the host organism, and we used to change the phenotype of the product of the host. It includes genetic DNA isolation, isolation of gene of interest, making of recombinant DNA, recombinant DNA, and transfer of this recombinant DNA to the organism. And after that, we used to receive the specific phenotype or product. The second principle is bioprocess engineering. The main principle behind this bioprocess engineering is just to maintain the sterile condition. It means to make the environment contamination free, and uh, this is the ambience environment in the chemical engineering process we used to create for the preparation of several products utilizing the life forms. It enables the growth of desired organisms in large quantity to produce several biological products in the production of antibiotics, vaccines, and enzymes, etc. It means we used to maintain the sterile condition to produce several biological products. So first step in genetic engineering is isolation of genomic DNA. And we will study what is the basic principle behind the isolation of genomic DNA from the plant cell. So first we see what are the processes we use to undertake to isolate the genomic DNA. First we used to crush and mince the tissue and make slurry of the lysis buffer. Means when we used to take the tissue of the plant either from stem, from leaf, or any part of the plant, and what we do, we used to crush and mince with a buffer which contains generally cellulase. Cellulase, what it does? It digests the cellulose and anionic detergent in buffer. So this buffer, what it does, it lyses the cell. The second step is separation of nucleic acid from protein by protease treatment. Then what we are doing, the tissue mixed with lysis buffer will be treated with the protease. So protease, what it does, it digests the protein. So what will be the remained product? That will be devoid of any protein. Then what we used to do, we used to degrade the RNA by using RNase enzyme. Then what we will get, after some uh, after treatment of RNase enzyme, some of the molecules have been removed, means some of the contaminating molecules will also be there. So we will remove by appropriate treatments and some chemicals. And what we will do, what we will precipitate the DNA by using ice cold ethanol. In ice cold ethanol, we will get the purified form of the genomic DNA. This is the very essential step, isolation of gene of interest. Means if I want to express a particular protein, then I need to get that particular fragment of DNA which ultimately transcribes and translates in that particular protein. It means if I want to express the insulin protein. So I need to get the fragment which ultimately translates into that particular protein out of the total genomic DNA. So there are several steps we need to follow to get that particular DNA fragment. So we will see one by one. First is restriction digestion. So what is the restriction digestion? We used to uh, use an enzyme called restriction endonuclease enzyme, which cuts the DNA at specific sites. It means out of whole genomic DNA, it will cut the boundaries of that particular gene. Then what we will do? We need to isolate that particular digested or cut DNA fragment out of the genomic DNA. So we will load that particular digested DNA into a gel-like platform, and it is called as agarose gel. This agarose gel contains ethidium bromide. So after application, and we used to employ the electric current. As we know that genomic DNA is having negative charge. So when we will imply the electric field, then the negatively charged DNA will move from negative uh, electrode to positive electrode. So what will happen? The DNA will start moving from one end of the gel towards the another end, and by this the genomic DNA will be separated on the basis of their molecular weight or the size of fragments. We can see here that this is the second number figure is showing the agarose gel. We can see here are several wells. We used to load this digested DNA, and we used to employ the current. We can see here we have loaded this DNA in the wells here nearby the cathode, and at the another end of this gel we used to employ the anode, and we used to supply this current. Then what happens? This DNA, the loaded DNA, will start moving towards anode, because as I said, the DNA is negatively charged, so it will move towards anode, means positively charged uh, electrode. Then what will happen? This gel, we can see here the details. The agarose gel is a natural polymer which forms a pore, means it forms a mesh-like structure. Several pores would be there, and that will help the DNA to separate on the basis of their size, and we used to extract the desired fragment on the basis of their size. We can see here that in this figure, the first figure, we have loaded the DNA, and after some time we can see here on the basis of molecular weight, they will run towards anode. So as we know that if several molecules are moving through a pore, then the smaller fragment will move faster as compared to the larger molecule. Then what will happen towards the anode side, means towards the lower side of this gel, the lower size of low molecular weight or low size of DNA will come, and the larger size of DNA or heavy molecular weight of DNA will move slower. So the different length of fragments of DNA will be separated. So we can see here in this third figure, we can see this is the marker DNA. Means we know the size of these each and every fragment, we know it, and these are unknown DNA sizes. So what we are doing in first well, second, third, fourth, fifth, and sixth. In six wells, we have loaded different digested DNA. So according to their size, they will move. The lower, we can see here, this lower, we can see here that the lower fragment is moving faster, and it will come nearby the lower side. We can see here the 250 base pair, 500 base pair, 750 base pair. It means when we will move towards the upper side, or we can say when we will move towards the cathode side of size of side of this gel, then we will find the heavy molecular weight DNA. So on the basis of this molecular weight, we will come to know that what is the region where my DNA, digested DNA, will present. Then what we need to do, we need to cut that particular site. Means suppose this is my digested DNA, and I know on the basis of this fragment that this is the desired, this band is containing my desired DNA fragment. Then what I will do, I will cut this agarose gel. We can see here that this fragment has been cut, and I will then what we need to do, we will collect it and we will put it in the tube, and by several uh, by employing several chemicals, we will isolate that particular fragment, and by this process we will get our desired fragment out of the total genomic DNA. Now we will see the details of restriction enzyme. As I said before, that this is a this is considered as the molecular scissor, and it cuts the DNA at specific sites into fragments. This enzyme protects, generally, this enzyme is naturally present in the bacteria, which protects the bacteria from their pathogens, means viruses. It means when viruses will infect the bacteria, then it is a kind of defense system in the bacteria that will digest, or that will cut the DNA of the virus into fragments, so that virus will be neutralized. So these are the enzymes, and are of three types. Means the on the basis of their characters, they are of three types. But out of those, type 2 has been used in genetic engineering or molecular biology experiments. So first we will see what are the properties of type 2 restriction enzymes. The first thing is it is an endonuclease. Endonuclease means it cuts the DNA inside the DNA fragment. It means it will not cut the DNA at the terminal end. Second point is it recognizes the cut DNA at the same site. It means it recognizes a particular fragment of DNA on the basis of their nucleotide, and it cuts the same uh, site and at the sugar phosphate backbone at a specific site of DNA. And the third point is it cuts a palindrome sequence. So we need to know what is the palindrome sequence. So for example, this is a sequence which is recognized by a particular restriction enzyme in the DNA. And what is the meaning of palindrome sequence? When we will invert it, we can see here that this is the fragment, and we have inverted this fragment, and what will happen, we can see here the sequence TTA, TTA, GCAC, and after inversion, the sequence is same. Now we can see here that TTA, GCAC. The same sequence will come when we will invert it. So what it does, it cuts the palindrome sequence. It recognizes and cuts the palindrome sequence. And for example, we can see here, for example, EcoR1. EcoR1 is one of the most important endonuclease we are using in genetic engineering experiments, and it recognizes and cuts the DNA at the site GAATC, and it cuts after G. And we can see here how this restriction enzyme EcoR1 cuts. We can see here that this is the DNA, but out of that particular uh, uh, out of the whole sequence, it recognizes a particular site which is GAATTC, and it is cutting at a particular site. And we can see here this is the DNA fragment. When we have used this EcoR1 restriction endonuclease, then we can see two different fragments, means it cuts the DNA into two fragments. And now we will see what is the criteria of nomenclature of this restriction enzyme. So first capital letter represents the genus of that particular species from where we are isolating this. The rest small letters is representing the species of that particular uh, 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 organism from where we are isolating this. Again, the capital letter represents the strain of that particular bacteria, and after that Roman letter comes, which represents the number or order of isolates. So as an example, we can see here in this slide, E represents the Escherichia and it is the genus of the bacteria. Co means coli. Coli means it is the species of that particular genus. R is the strain type, and first is the first identified, means order of identification in the bacterium. Now we will see what is ligase enzyme. We will discuss it later. What is the use of this ligase enzyme? Generally, it seals the gap in DNA fragment. It is used to join two different fragments, and these are isolated from E. coli as T4 phages. Thank you.